It's my pleasure to here present my research work today. I'll talk about the Golgi targeting at first and then present a new method of cancer therapy. We call it the mechanical disruption membrane for cancer therapy, or MDMDV therapy. My lecture will start off with a short introduction by stating why we target to the Golgi. Then have two examples for the Golgi targeting, and at last go to the summary and the perspective. As you know, um, Golgi is one of the major organelles composed by serious C-cells. There have been lots of researches about the structures, biofunctions, and biological significance, even though there's still a lot of arguments on certain points, such as origin of Golgi or the relationship between the Golgi structure and function, etc. In order to set these battles, basic requirements such as high performance image or effective targeting are needed. In this high performance fluorescence image, organic dyes such as silver iodide derivatives, gene engineered chromatography such as fluorescence proteins have been widely employed. However, these days has problems such as for the ceramides diffusing cells and can target to the Golgi by need 2 hours and are easy to be photo bleach, or the fluorescence proteins are too big to act as a target motive. So in such case, we developed fluorescent carbon dots, which can be used for long-term imaging of Golgi in living cells without significant photo bleaching. By hydrothermally mixing diaminobenzene and aminodiamine, we obtained the 3 carbon dots with the emission of yellow, green, and red. And it's very interesting that we find the yellow carbon dots when it was under the excitation of UV light, it has a blue emission. But if we have a yellow emission, if we immerse in a polymer solvent, it was found that these yellow carbon dots successfully targeted to the Golgi of various cells and emit blue emissions. This targeted to the Golgi could be identified by co-localization experiment with commercial Golgi tracker or etc. In such case, by using carbon dots, we can have very good observations of morphological fragmentation of the Golgi in physical processes, remarkably in the cell mitosis, and some characteristics inspired process such as apoptosis, which introduced under physical or chemical conditions. In order to identify the targeting mechanism, we posted the free amylose terminals with folic acid. As you know, folic acid binds with the receptor of cancer cell membrane, making the carbon dots cannot target to the Golgi, highlighting the importance of the basic substructure of carbon dots with amino groups for the image of for the Golgi apparatus. In order to identify the roles of amino groups, we prepared different types of carbon dots by using different substrates of amino or aminoadenine precursors. By changing these substrates, we obtained carbon dots 1, 2, 3, 5, 4, 6. As you can see, the carbon dots 4 and 5 and 6 which were prepared with the precursors without the amino groups, cannot target to the Golgi. And these prepared with precursor with the amino groups such as carbon dots 2, carbon dots 3, and 5 can target to the Golgi without exception. That is, the free amino groups on the benzene and the amine were equally important to the formation of basic structure and that can target to the Golgi. It's interesting that the yellow carbon dots have emission dependent on the polar medium, thus and can be used for imaging change of polar of micro environment of the Golgi. Since the membrane structure of the Golgi is composed of phospholipids and some hydrophobic molecules, and thus has a relatively low polarity microenvironment. So, with the polarity sensitive yellow carbon dots get entrance into the Golgi through fluorescence emission would be obviously shifted with the cell growth. That is, the polarity switch of Golgi apparatus during cell apoptosis was successfully monitored. An interesting finding is that the emission of the carbon dot 5 cannot shift, but sensitively decrease with the increase of polarity. So we can selectively develop the amino substrate carbon dots that are polarity sensitive and Golgi targeting image. On the basis of the finding of the free amino groups can effectively target to the Golgi, we further considered how about other groups such as multiple precisely to some specific atom and try to discover the role of live atoms such as sulfur during the targeting process. In the next, we will find that some amino acids residues such as cysteine are effective to the Golgi targeting on its sulfurhydryl or cell group. Our strategy is to prepare carbon dots with cysteine residues and assess the targeting ability in terms of its count, amount, chirality, and location of cysteine residues. At first, we prepared the L-type carbon dots by using citric and L-type cysteine through the pyrolysis process. As prepared L-type carbon dots is in the range of 5 to 12 nanometer and has core shell structure, high blue emission, and high photo bleaching. IR spectrum showed that as prepared carbon dots have cell growth, which obviously come from the cysteine residues. Interesting, if we use the D-type cysteine also as a precursor, a D-type carbon dots could be available, suggesting that the free C-on group gets preserved from these precursors. In order to preserve the functional group, temperature should be well controlled. Temperature gradient data taught us that the temperature of pyrolysis should be lower than the decomposition temperature of cysteine. Thus, we proposed the FGP strategy in order to prepare carbon dots with the active or functional group of the cell on their surface. The as-prepared L-type carbon dots through the FGP strategy were identified to specifically target the Golgi by studying HEP2 cells and other cancer cells with Golgi GFP or polypyrrolidine, showing that the accumulation of the L-type carbon dots in the Golgi has rarely occurred. Immunoassay showed that the carbon dots can stain both the size and the tri-side of the Golgi, while golden-doped L-type carbon dots TM images showed that their type carbon dots are mainly inside the lumen of the Golgi. These observations indicated that their type of carbon dots have a high specific targetability to the Golgi. It was found that the carbon dots has a very good photostability, even if under the emission the radiation of light more than 60 minutes. The intensity only decreased to 80%, indicating that the air-type carbon dots have a very good photostability, which was much better than those such as the polypyraceratic and the Golgi GFP. Cell treatment with a typical dose of air-type carbon dots didn't affect the quantity of secreted proteins, glycoproteins, and apoptosis proteins. And this DARC demonstrated that the air-type carbon dots couldn't generate reactive oxygen species during the long-term in situ Golgi image. Cellular uptake and intracellular transportation tests showed that the cellular uptake of L-type carbon dots are energy-dependent process, which was mediated by the Vebleo, cavity and clearing the intracellular trafficking of L-type was also studied by analysis the overlapping between the L-type carbons and different subcellular organelles at a different time. For example, at the third minute, L-type carbon dots had a relatively high overlap ratio with the easy endosome. At the 112 minutes, the number of L-type carbons were in the net of endosome and then lysosome. At 240 minutes, most of the L-type carbons are transported to the Golgi. Thus, the uptake path is mainly mediated by the cavity and cleavering, and a large quantity of L-type carbons are transported via early endosome, late endosome, and lysosome, and eventually to the Golgi. In order to understand the mechanism of this energy-dependent process, we prepared different types of carbon dots by using different precursors. At first, we considered the stereo configuration. It could be found that the L-type carbon dots were much easier to target the Golgi than D-type carbon, indicating that the chirality of the carbon dots strongly influences the targeting ability. Carbon dots prepared from precursors without chiral center are found to randomly spread in the cells, reinforcing the significance of a proper stereo structure of the carbon dots for the Golgi targeting. And second, the existence of free cell groups is essential for Golgi targeting. Carbon dots prepared from precursors without free cell groups are found to have very low Golgi targeting ability. These observations separate the citric role of the free cell groups for the Golgi targeting, and also suggests that the L-type carbon dots bind the cell group receptor site of the Golgi, which might be through the formation of disulfide bonds in the exciting environment of the Golgi lumen. At last, we should consider the cysteine group. As to the targeting of L-type cysteine, we choose chemically modified organic small molecules and nanoparticles with the L-type cysteine, and then incubate with the HEP2 cells. We can see that the fluorescence without cysteine functionalization is found to readily spread over the cell, over the whole cell, while L-type System functionality fluorescence can fully co-stem with body pack ceremony. Similar results could be observed for the system modified 40 and 80 nanometer silicon nanoparticles, further stressing that the system residues are critical to the targeting. Furthermore, we found that the targetability of the molecules such as TCPP is dependent on the linked number of L-cysteine residues to the Golgi. TCPP without any cysteine modifications spread over the whole cell, while single system functional TCPP can localize with cells with Golgi GFP after 5 hours incubation. The double nesting functionality TCP needed only 3 hours to match with the Golgi GFP, suggesting that the Golgi targeting ability is closely associated with the number of L-type cysteine residues, showing that a multivalent effect of L-type take effect and greatly improves the Golgi targetability. That is, L-cysteine is an effective agent for specific targeting to the Golgi. In such case, we expand the capability of the L-type carbon dots further for Golgi targeting. At first, we can see that the L-type carbon dots stem cells can precede normal proliferations, indicating that the L-type carbon dots are very biocompatible and can be applied for the long-term image. Second, the Golgi is sensitive to virus invasion. By visualizing the dynamic morphologic changes of the Golgi apparatus during the virus infections, we can see that the Golgi apparatus HEP2 carbon dots stem cells have blue fluorescence emissions. With the infection of virus, the blue fluorescence gradually collapsed, indicates that the Golgi apparatus was fragmented. Cells were found dead within 12 hours. Since the rapid collapse of the Golgi apparatus, thus we suppose that a close relationship between the collapse of the Golgi apparatus and the death of the cells, providing us with the new mode for investigating the virus infection in early stage and a new stage for antiviral drug development. Since collapse of the Golgi is an important reason of cell death, so we considered how to make use of the collapse of the Golgi for the cancer therapy. Here we propose a new method called mechanical disruption of membrane for cancer therapy, or simply as MDM cancer therapy. As you know, the most popular strategy for drug development, including those for immunotherapy, antivirus, and antibiotic, are based on this basic interaction between the ligand and the receptor. Owing to the richness of variability of life activities, though the receptors as the target of the drug might change, making it hard for drug to find the binding site, decreasing the treatment effect, and led the occurrence of clinically so-called acquired drug resistance. Even if not with efforts, including new chemical structure, or new pathway, multi-pathway, or nanotechnologies have been taken, the acquired drug resistance still existed because of the ligand receptor binding mechanism doesn't change. In such case, we consider by taking care of the cancer therapy as an example, we try to resolve the problem of acquired drug resistance by following two proposals. The first one is to change the binding site, namely the single receptor as a subcellular organelle, which can avoid the change of binding site. The second is the one to change the small molecule as assemblies, which have mechanical disruption capability. In such case, we developed subcellular targeted mechanical disruption for cancer therapy without drug resistance through targeting, delivery, and assemblies. The best route is to deliver an effective mechanical disruption motif to the gold, wherein the motif starts a supramolecular self-assembly to form a mechanical structure, making the membrane of the Golgi ruptured. So we call our method as a mechanical disruption of a membrane for cancer therapy, or shortly as MDM cancer therapy. Our method started from the finding of the mechanical disruption motif. Our eyes concentrated on the self-assembly of short peptides. By testing lots of peptides, we found that 6 peptide L-type F4KY is a good candidate, which can self-assemble into a left-handed nanofiber with a beta sheet. An interesting finding that the D-type F4KY cannot self-assemble into any type of structure. Why the L-type F4Y can form a left-handed helical fibers, while with unusual beta sheet, and the D-type F4KY cannot. By using a nuclear oversaw effect spectroscopy, we found that the phenylalanine amino acid residue between the two L-type 6 peptides have strong interactions, and the proton's distance smaller than 5 angstrom. That is, the space hindrance makes the D-type short peptide not able to interact with each other between the two type 6 peptides. There are 4 interactions between their type 6 peptides, including hydrogen and the partner zipper, pipe interaction between the benzene ring, and the electrostatic interaction between the hydrophobic end group. These interactions merge the L-type 6 peptide self-assembly into an L-type helical fiber, which has unusual beta parasite. Computer simulation shows that unusual beta sheet, parallel sheet is not generally the anti-parallel, but the one of the parallel. In order to identify the assembled left-handed helical fibrils have enough mechanical capability to disrupt the organelle membrane, we measured young monomers of the assembly of different helical peptides, including F4DKY, F4KDY, and found that only the F4KY has the largest young monomers. That is, our left-handed helical fibers have strong rigidity to disrupt the organelle membrane. By constructing a multi-aminar vessels, we made a series of net tests and showed that these non-physical structures cannot easily disrupt the membrane of multi-aminar vessels. However, the one of L-type 4KY, if released through the cleavage of furan, was assembled in test tube and seriously destroyed the multi-aminar vessels, indicating that left-handed fibers could effectively disrupt the membrane. In order to understand this progress of membrane disruption, we made a molecular dynamics simulation. Here, the brown represents the water, and the brown and the blue is the membrane, and the green is the assembly. And we can see, owing to the entrance of helical fiber into the membrane, the fiber starts bending and extruding the membrane and forming a big water channel. The water channel might reach 3.6 nanometer. If such a huge water channel is in cell, it will produce a huge disorder of ion and molecular activity, showing that the mechanical action of spiral fiber can promote the cancer cell apoptosis. Furthermore, tubal tests showed that the disruption of ability of helical fiber is far bigger than that of non-bigal fibers formed by these conformational isomers, and also far bigger than that of spherical particles. As I stated before, sensing uh, was very good for Golgi hacking. So sensing gold peptides were applied for deliver the assemble unit. Experiment showed that six sensing peptide is the best one. Immunoassay image showed that the cancer cells can highly express transferrin receptor and furin, and this sensing oligomer peptide was contributed on both sides and trans sides of the Golgi, particularly the side with a much more transferrin receptor, which was identified by inhibiting the Hansen expression of the transferrin receptor. Owing to the high expression of the transferrin on the surface of the cancer cells, the six-cysteine oligomer, cysteine, therefore was relatively enriched in the Golgi apparatus of cancer cells. As I stated before, cysteine was the targeting and the assembly process could be made with the size of freight and a signal by combining with these units of targeting, delivery, and assembly together. A 16 peptide composite, which was composed by targeting unit of 6 cysteine, furin restriction unit of RVRR, which coupled with freight donor and acceptor K, and then assembly unit of L4KY. For dynamic tracking, fluorescence donor acceptor at the two ends of the cleavage substrates of furin during the delivery. The appearance of freight makes the fluorescence signal off, so that makes the delivery could be easily observed. In order to identify our design, we made a series of tests. The first one is the release of the assembly unit through the furin restriction. As you can see, the assembly unit forms nanoparticles without furin, and the presence of furin release the assembly unit and forms helical fiber. If the release occurs in cancer cells, green emission named freight donor was clearly observed, indicating that delivery and release of the assembly unit was successful. And further, immunofluorescence assays showed that the assembly unit was released mainly by the trans of the Golgi, and the flow cytometer showed that the composite by the 3 parts can successfully recognize the cancer cells. The spectral furin cleavage was first identified as a characteristic mass peak. SEM images showed that incubation of the 16 peptide composed with the cancer cells, F4KY, released in the Golgi and assembled into fibers and destroyed the cancer cells with the time from start 4 hours and 8 hours. Further tests showed that the cancer variability of cancer nails, not the normal cell nails, was drastically affected. Immunoblotting and cyclin secretes analytical results showed that major reduction in the spontaneous section of cyclotime was observed after 80 hours incubation, indicating that apoptosis and neurocrisis, two completely different cell death processes, may both reduced after the assembly of the F4KY. Because of the mechanical disruption and induced the multiple death pathway, we suppose that acquired drug assistance can be avoided. In such case, we tested whether the breast cancer or cells, and some animal test. And a significant shrinkage of tumor tissues were observed. However, the major organs, including the heart, kidney, and the others, cannot be affected. So the cancer tissue tests are obviously empty and compared to the untreated one. In such case, we can conclude that our strategy of MDM cancer therapy was successful, namely 6 peptide at the N-terminal was designed at first, and then used to differentiate between cancer and normal cell lines, and then a tetrapeptide RVRR, which was a substrate of furin, and with the cleavage of the furin. The heptapeptide F4KY was released and itself into a left-handed conical fibrils. They are the assembled left-handed clinical fibrils, then chemically disrupt the Golgi apparatus membrane, resulting in structural damage and biomaterial disorder, and eventually reduces cell death of cancer cells without required drug resistance. In such case, I concluded that we construct a new strategy to target the Golgi, which was dependent on the amino groups and free cell group, either on the surface of carbon dots or connected with oligomer peptides, making the cysteine residues as a good candidate to target the Golgi. And with the delivery of cysteine groups and furin expressed in the Golgi core cells, a 6-peptide F4KY was released on the crans side of the Golgi, self-assembling into helical fibers and mechanical disruption the membrane of the Golgi, making the new cancer therapy based on the mechanical disruption was developed, which showed less acquired drug resistance. Our strategy is not only about killing cancer, but also importantly, this organelle targeted mechanical destruction concept may be used for solve or reduce the problem of bacterial resistance and virus resistance. The mechanical action leads to large-scale function and structure loss, and is basically not affected by a small number of targeted mutations or pathway changes. Here I greatly appreciate the financial support at my university, my lab members. I deeply thank the organizing committee and your attention. Thank you.